soon we won't have to actually have meetings, or we'll just live stream. Yeah, make it look more full. Okay. This is hello? Yeah. Okay. Great. So let's get started. It's almost the academic hour is almost started, 10 after 12. So it's my pleasure to introduce Naomi Leonard, who is the Edwin S. Wilsey Professor of Mechanical and Aerospace Engineering. One thing about the MBD is you will always have people doing things that you would never normally be exposed to, at least in my case. She is her main area of interest at Princeton is as a control theorist who basically studies how complex systems, signaling within complex systems drives collective motion and decision-making in biological systems and engineering systems. She applies a lot of her work at the level of animal groups, but also has some interesting work maybe she'll talk about robotic vehicles in Monterey Bay, California. So if you need an assistant, please come and talk to me. Does she? Oh, you have one. Okay, great. Thank you. Over to you.
Thanks. So thanks very much. It's a pleasure to be here. So I'm going to talk about resilience and spreading processes, which are, I think, you can think about in a lot of different domains. I came to it through thinking about the spread of behaviors in animal groups in motion. And I've been fascinated for a long time now by animal groups, not only because their motion patterns are so spectacular, visually spectacular, but also because they exhibit a remarkable kind of resilience to challenging environments that change over time. So by resilience, I mean the ability of the group, and not necessarily a lone individual in the group, to be both responsive to environmental cues and yet robust in what they have to do with respect to uncertainty and disturbance and perturbation. So we, in my group, work with analytically tractable and generalizable models to try to uncover mechanisms that explain how individual animals are responding to both social and environmental cues, and how this leads to the kind of resilience that we see at the level of the group. And the analytic tractability is super important because it allows us to think about deriving testable predictions as a function of a few critical parameters in a systematic way. And the generalizability then allows us also to think about how the sort of mechanisms that we start to uncover might be relevant or suggestive of what might be going on in more than one species. And then, as Jeff mentioned, both analytic tractability and generalizability allow us then also to think about the design side. So in particular, in technological systems, how we can think about deriving dynamics that would allow these non-biological systems to inherit some of these amazing features, if we can understand what's leading to them. And I have done a lot of work in thinking about networks of robotic vehicles as sort of new ways to think about observational systems in the ocean. So moving in patterns and doing things that fish schools might be doing, or other kinds of animal groups, but for the purpose of we work with oceanographers and ecologists to collect data in new ways. So I want to sort of convince you of this sort of model plus data-based approach to things and thinking systematically of things. And I want to first just sort of talk briefly about sort of a range of scales of animal groups and the kinds of things one can say. And then I want to sort of focus in on this problem that I advertised that I'm going to talk about, which is this investigation that we did recently of resilience in the regulation of foraging in red harvester ants in the desert of Arizona. So let me just start with this beautiful video, which maybe you've seen before, but here are more than 1,000 starlings flying over. This is the Rome train station. It's in the winter, in the evening, and one thing you can observe is, I think you saw there's a predator there, and they seem to be doing a pretty good job of moving with respect to the predator as a whole. And yet at the same time, I mean, despite looking at them, they seem to move so synchronously, almost immune to what must be an enormous amount of, uncertainty about what everybody is doing around them. And so it's these kinds of questions. We actually collaborated with the scientists in Rome who filmed these. They're actually physicists. And to ask these questions, systematically with analytically tractable models plus their data, to ask questions. In this case, we looked at the problem of understanding the role of network structure in their dynamics. So here, network structure is describing who is sensing and responding to whom, and what role that plays in the ability for them to stay, so uniformly together despite uncertainty. So I've likewise done a lot of work thinking about fish schools. Here's another beautiful video. This one I just took from the web. And again, what you'll see is it looks like this group is, as a whole, is responding in a very sensitive way to some cues in the environment, because they move from this kind of circular motion to a sort of a polarized pattern. And yet, there must be so much uncertainty, and again, they seem so sort of immune to uncertainty and disturbance because they move so synchronously. And, so again, I've been working over many years, over a decade, with people like Simon Levin at Princeton and EEB, and Ian Cousin, who used to be at Princeton, now at Max Planck, who runs experiments with schools of fish to try to tease out mechanisms that explain how they can be so responsive. And in this particular paper we published several years back, we were looking not only at network structure, but what happens as a result of changing network structure. So fish move relative to one another, and they change who their neighbors are, and how can we understand that as a mechanism that might explain, for example, how only some may know something about an environmental signal, and yet they're able to distinguish and deal with conflict and these kinds of things, figuring out, in this case, consensus decisions about where to go. We've also been looking, again, with tractable models together with data, and thinking about, motion that's associated with collective decision-making. And this is a very famous and spectacular story of house hunting honeybees. And so it's very well known that honeybees are able to make decisions when they're looking for a new nest site that has to serve them well over the course of the winter, how they pick the best quality nest site as a group. And the question we asked, and it came about from a paper that Tom Seeley and company published in Science, again, several years back, that not only can they pick best of N, but they're also able to handle the flipping the coin when they are confronted with near equal quality nest sites. So unlike the U.S. Congress, where they would probably debate forever and ever, nothing would get done, they can't, and so somehow they're able to flip a coin. And so it turns out that, it's already very well known that the way that they're able to, first off, pick a good nest site is using this advertising that's called the waggle dance that they do on the vertical part of the swarm. So there's something like 10,000 honeybees sitting here, and 300 or 400 of them go out, scout out sites. They come back and advertise, and they can advertise where and how, of what quality the site that they have just explored. But it turns out that there's another signal called the stop signal, which is sort of a gentle way of talking. It's like the shut up signal, because one who's dancing for one site is getting head-butted by the other who's committed to another site, and it has this effect of tempering the dance. And we show with models that this allows them to do this. It's actually value sensitive decision making when you have near equal value sites. And so again, we develop models and try to understand how to explain these kinds of behaviors, and then, these are generalizable models. So we move on to thinking about sort of more abstract ways of exploring other questions, like, again, what is the role of network structure? What's the role of individual differences? There might be heterogeneity in these systems that is playing a big role. So these are, again, things that we want to ask in systematic ways. Here's another where we actually set out expressly to think about heterogeneity. This is in heterogeneity in these herds of zebra that are evading a threat when they have different levels of agility and speed. So there are mothers and foals. There are males in these different groups. This is actually data that was collected by my former student Will Scott with my colleague Dan Rubenstein in Kenya. This is actually, the threat is a little robo-lion, so it's a little wheeled car that Will is driving, and they're afraid enough. But the goal here was to try to, again, use the data, develop these analytically tractable models to try to understand the relationship between social cues, which keep them together, and evasive signals from this threat. To try to understand, they don't do this spreading apart, they stick together. How are they doing this? How are they so efficient, actually? They don't run when they don't have to. And how do they organize themselves? All kinds of interesting questions when they're in this group that's heterogeneous. I also just wanted to mention that we think about humans, and I've been thinking about humans in the context of dance, because it's a kind of dancers have been trained to be highly physical aware of what's going on around them. And so this is actually a little experiment at the end of a project with my colleague Susan Marshall, who's a choreographer, and thinking about what happens when dancers employ the rules that we use to model schooling and flocking. We also had a more recent, so these are all dances that are, so you call it structured improvisation, so they're rule-based dances. So you can actually sort of run experiments with humans and try to understand some kinds of phenomena. But in the second piece, this was actually part of the creation of the dance, and this was a piece that was premiered in New York in 2016. But we also, if you're interested in this combination of art making and sort of social decision-making dynamics, we actually use an evolutionary dynamic model to try to understand this in the making of the dance. And so he recently published a paper on this stuff. I'm not going to show you this stuff in the ocean. We do a lot of work in the field, but we also do a lot of work in the lab. This is just a little a look at sort of the honeybee story has grown into one of decision-making among agents and thinking very hard about how to think about it systematically. So this is just a little sort of a visualization in my lab of two individuals that are trying to choose between the right side of the room and the left side of the room. And this story is that we can try to understand it in terms of two parameters, C and U, and C is the color in the room, so they're measuring the colors, and we're using color here. Could be could be temperature, could be all kinds of things. What is sort of representative in what they did in this setting was we showed how they would transition from agreeing on A to disagreeing, and you could even think about it as like a task allocation problem, and something changed, and so somebody had to go here and somebody had to go. So taking on different kinds of roles in this tiny network of two. Okay, so in all those cases, I sort of went through it fast. The idea was we use these analytically tractable models to try to, together with data, uncover mechanisms and come up with testable predictions and go back, but also then to move on to generalize these models to see what kind of larger questions we can ask, and then we can kind of move between different kinds of model problems, not to say that the mechanisms are the same, but some of the underlying sort of mathematical structure might be useful in other settings. And so I want to kind of tell that story, but through a particular example, and this is this recent work that I've done with my student who graduated in July, actually, Renato Pagliara, and Deborah Gordon, who's a field biologist. She's a professor at Stanford, has been studying these red harvester ants in the desert of Arizona for more than 30 years. So this is the story of resilience in their regulation of their foraging. So these are ants that collect seeds that have been blown around by the wind. So they do not leave pheromone trails. Instead, they go out on a trail to a big foraging area, and then they spread out and look for a seed. Each one finds a seed, comes back actually along the same trail, drops a seed. And what's interesting is that, so they don't have any way to communicate by leaving pheromone trails. Instead, the ones after they drop their seed, they greet some of the available foragers that are sitting in the nest, and they exchange a cuticular hydrocarbon. And so what happens is that the ones sitting there can sense whether they've just greeted somebody who's actually been outside, and they can also sense if they've been successful at finding a seed. And so it's, what Deborah has shown is that they essentially get excited. The more individuals that are coming back, the more interest that they get in potentially going out. So she sees that they're more likely to go out, the higher the incoming rate. But what's super interesting, and this is sort of the big question that she actually came to us to ask, is that there's this important dilemma that has to do with water. And that is that they collect these seeds, and they're metabolizing the fat in the seeds to get water. But on very hot, dry days, it's pretty dangerous for them to be going out because they can become desiccated. So they have a sort of water trade-off. And the question is, what are they doing? How are different colonies distinguished? Because what she's found is that, that the colonies that show the most reproductive success, so she knows whose daughter and granddaughter colonies, and she can see strategies that are surviving and strategies that are not, about how overzealous or underzealous one should be going out on a particular day, that the ones that are foraging less on hot and dry days have higher reproductive success. So there's this notion that their ability, their sensitivity and responsiveness to this environmental condition may have something to do with their fitness, right? So our big question was, how is the water trade-off managed? And managed over minutes to hours, and particularly this sort of sensitivity and responsiveness to temperature and humidity, because she had only been really studying kind of what was coming in and out of the nest and not really thinking about the full closed loop system, because of course the guys that go, they're all she's, that go out are the ones that come in, and you have to think about that whole loop if you're going to think about minutes to hours. And what are sort of key parameters of this system that determines these responses, governs behaviors, and in particular can distinguish these colonies? What can we even call a strategy? How do we distinguish strategies? And so we wrote this up, and I'm going to tell you this story, but then we also went on to this sort of, how do we then generalize? How do we abstract out these notions? Because if you think about it, it is almost like an infectious process, right? They're becoming infected with this foraging behavior. And so we went on to think about how to generalize this notion of resilience, because the ones who are really doing a good job are the ones that are being resilient to changes in temperature and humidity, to other kinds of aggregations. And so we also wanted to explore, I'll show you that when we first studied this, we looked at a sort of more of a population level model, and we wanted to think more at the level of, sort of agent-based stories. How do we understand things like the role of individual differences even within groups, or the role of network structure, like who actually is interacting with whom and what role does that play? And then also, are there sort of, because we're doing this in a very systematic way, are there ways to try to understand systematic strategies for control or intervention, like to either enhance or to stop some kinds of spreading? All right. So let me just begin by showing you some of the data that motivated us. So Renato went out with Deborah, so we have data from 3 summers. It's sort of end of August, beginning of September, that she goes every year and collects this data. This is after sort of a lot of rain. And then they go out and collect the data, and the data is literally timestamps of ants going this way and timestamps of ants going this way, and then you can filter that and get rates of incoming and outgoing ants. And so what you're seeing here are just 2 different colonies from August 20th 2016. The blue line is the foraging rate in ants per second over the course of the morning. So they come out early in the morning and then they go in by 12:30. It's so hot, they all come in. And in fact, she doesn't study them in the afternoon because all the researchers come in, it's so hot. But there's all pretty interesting things to observe. First, that the outgoing rate is higher than the incoming rate at the beginning, which you might expect. But then they actually kind of equilibrate to a rate where the incoming rate is equal to the outgoing rate. So we think of that as sort of a quasi-steady state. But then the other thing that's super noticeable is that, this is the exact same day, exact same temperature and relative humidity, and this colony is foraging at a rate that's more than twice the foraging rate of the other one. So that's pretty extraordinary. And so here's just some other examples. So this is actually the same colony on 2 different days, okay? And you're seeing again, blue is incoming, red is outgoing. Green is kind of like the integrated of the difference. So it's sort of like how many ants are out there. Yeah.
How far are the colonies from each other?
That's a good question. Yeah, they're I don't know in meters, but there's.
But
There are hundreds and hundreds of them. Yeah, she studied that whole story of territorialism and things like that. But
is that a factor that controls?
They don't. They really don't breach each other's kind of arenas.
But in terms of their like the location relative to food sources or something like that, there's a higher density of.
I think they get blown. The seeds get blown pretty uniformly. Yeah, but those are all good questions. Yeah. So you can kind of see again this pattern, like the outgoing rate. In fact, is pretty high relative to this, like a big transient here. But even this colony on 2 different days, so clearly this is a hotter day and a less humid day, and the rate has gone way down. So this is clearly, well, it seems to be a nice illustration of their ability to assess what's going on outside in terms of temperature and humidity. This colony on September 1st. In fact, the temperature and the humidity is about the same as this one. It didn't even stay out there. It basically went out and came back in, as if they assessed this as being not a good day to be foraging, whereas this one thought it was a perfectly fine day. This plot I just want to show you because this is pretty interesting. This was just sort of a replotting. Now I'm looking, instead of like these foraging rates as a function of time, this is the outgoing rate as a function of the incoming rate. Right, so you can see that the outgoing rate is above so this horizontal. I mean this is the line when the 2 rates are equal, and so outgoing rate, and this is time of day. So the blue is earlier in the day. You can see it's much higher than the incoming rate, and then it equilibrates, around this line where the 2 rates are equal. Here it does the same kind of thing, but it's just at a lower rate. Here. It basically doesn't really spend much time there. It just goes in and comes out, right? So these are this is actually a little bit easier to kind of distinguish these 3 different kinds of behaviors, right? So the first question was, well, let's think about how do we model this? Our first idea was that, and it wasn't just our idea, so we were leveraging some work she'd done we're actually working with some neuroscientists thinking about models of neurons as excitable systems, right? Because it's really an excitable story, right? You're getting excited by these interactions, boom, and you're like, I'm going to go out and forage. So the first step was to think about what I call the open loop, which is sort of how do I model these guys coming into the nest? What's the map from incoming to outgoing for the nest, right? And so we model this in 2 little stages. One is, the map from the incoming. So, and they're interacting to some kind of stimulus, which I just call S, right? And so we actually just model this simply as a leaky integrator or low-pass filter, whatever your preferred language is, the same thing. So this idea that, you get a jump up in your stimulus, but you forget it after a while, and that would be the time constant associated with that. Here is called tau. And then we have a second stage, which is like, how do you then respond to that stimulus? And we use, this is sort of straight out of neuronal dynamics. We use a model. This is sort of like a low-dimensional model of like the Hodgkin-Huxley equations for the dynamics of a neuron. So this is essentially a nonlinear oscillator, and we sort of normalize it in a way that the parameter that we really are going to think about as something that we want to understand is, we've just sort of picked one that allows us to think about C, this parameter, as the susceptibility, and higher C means higher frequency oscillations, and we interpret an oscillation, like the positive part of an oscillation crossing a threshold, as an ant going out. Okay, so essentially, if C goes up, if there's, they're more susceptible to these interactions, we see, a higher outgoing rate. Okay, so we fix a lot of the other parameters just so that we can have kind of the whole range of dynamic behaviors, and we focus on C. So if we think about this idea of analytic tractability, getting models that depend on only a few parameters, C is the first one that I want to introduce to you. Okay, so this just gives you a little sense of what a profile might look like. So here's kind of these spikes corresponding to ants coming in. I got a spike every time an ant is coming in. It goes through this first leaky integrator, because those are the interactions, and you can kind of see that the stimulus, we get these peaks, and then they decay, and when they come at the same time, you get a higher peak, and then that decays, and so that can reflect some of the crowding in the nest area, which is actually something of importance. And then it goes through this nonlinear oscillator, so when the stimulus is high enough, we get oscillations, and then when it's low, we get, we don't get oscillations, and this parameter C is the key one that would tune, the frequency of these oscillations, and then, this goes through that thresholding, and we get our R out. So this is a way to think about R in, the rate of incoming ants, to R out, or just the spike, the lambda in to the lambda out. So we can do all kinds of analysis already, right? So we can, make some assumptions, like maybe this is a Poisson process coming in with mean incoming rate. So then we can draw some conclusions about the stimulus being ergodic. It's going to allow us to make some conclusions, like in long term, time statistics will converge to ensemble statistics, so that we can actually do things like approximate the probability density function at steady state of the stimulus being at a particular value. We can also, we use the timescale separation. There's a timescale separation I didn't point out in this model to help us approximate, as a function of C, the frequency, the period, the limit cycle, the cycling period of this oscillation. So we can actually analytically write down an expression for the map from average incoming rate to average outgoing rate. These B's are sort of the smallest value of the stimulus that gives you oscillations, and then there's a max value of the stimulus that gives you oscillations from the Fitzhugh-Nagumo dynamics. And so we can do things like this. And so this is analytic. This is our approximation of mean outgoing rate as a function of mean incoming rate for different values of C. You're already starting to see the kinds of curves we saw in the original data when we replotted them on the right. And it's kind of interesting. One thing to note is that, well, you can't really see it, so I'll just tell you, like for these two small values of C, this is sort of maybe tangent to this line when the slopes are equal, but it never goes above it. You never get kind of more ants going out for a single ant going in, which is going to lead to something really interesting. It's going to lead to the fact that you're not going to. It's going to help us understand those situations where you don't equilibrate on that line. This is just sort of showing you that these are just simulations, just showing you that we're getting pretty close. There's a little underestimation of the period. So I mean, these peaks, I don't know if you can see, are a little bit underestimated. We understand why. Yeah.
Just to understand, so the two axes just are the U and V variables in the Fitzhugh-Nagumo. The U and V, the two axes here.
No.
So what is getting mapped to that?
The axes are this average of this sort of average rate going in and the average rate going out, right? So we have the dynamics of the stimulus, and then we have you were talking about V and U, yeah. So we're decoupled from that. So the V is the one that we care about, right? That's sort of the voltage, and this is like a gating variable, right? But these are you know, two different timescale separations, right? So this is slow relative to this, and this is slow relative to this. Okay, but what we can also sort of surmise is that this good agreement suggests that this convergence to ensemble statistics actually happens at even slower, you know, shorter time. We don't have to wait forever to use this approximation. Okay, so now the question is, let's close the loop, right? Because the ants going out become the ants coming in. So I just talked to you about mapping incoming to outgoing. How do we map outgoing to incoming? Once we've done that, then we've closed the loop. And that's just with this little box that I'm calling foraging. And we modeled this pretty simply as just kind of a time delay, right? And Deborah has a lot of really good statistics. So she basically told us that you could model it well with a chi-square distribution. So capital D is yet. So now I'm introducing a second parameter, capital D, which would be, you know, so the average time that an ant spends outside looking for a seed. And this is just sort of what these chi-square distributions look like. So this sort of distribution of foraging time, you know, when D is equal to 5, they're sort of, you know, left-skewed distributions. Okay, so we can again do something analytic, and this time we use something called queuing theory, right? So to find these analytic expressions now for the map from R out to R in, right? And we can use this kind of iterated map, and we can show things like that the solution when R in is equal to R out is actually a stable solution. So that means it's going to be robust to noise and uncertainty and perturbations nearby. This is a plot that we can also, this is also kind of analytic. This is the actual value of the quasi-steady state foraging rate as a function of C, right? And what I was hinting at before is that below some critical value, they won't go out at all. And that corresponds to the fact that some of those, for when C was too small, it didn't actually get on the left side of the curve. So this is just some, you know, so we know what this tells us, that together with what I showed you with the open loop maps, is that now instead of just being looking at constant in, constant out, this is actually a simulation of what would happen for a different value of C in the closed loop setting. And you see these kinds of behaviors where, you know, it starts out going above, and then it equilibrates on the line. And what you don't see is the fact that when we tried it, you can't see that for the small values of C, it's just sitting right there at the origin, right? This is just trivial foraging, maybe a little bit going out, but not much. But of course, this isn't quite some of those loops went up and down, right? And I also haven't told you about anything about assessing the environment, the temperature and the humidity. So this is the next step, and this is a hypothesis that was made based on just being out there in the field and recognizing the fact that the nest temperature and humidity doesn't change every day. So they wake up, you know, they come upstairs, and they don't know what it's like outside. So this was the hypothesis that they have. Basically, we're taking all the ants in the nest and splitting them into two groups. And at the beginning of the day, they're all in this group, which is sort of the uninformed group. They haven't been outside. And we're keeping track of ants as they go outside and come back, and then they move into this group, which we're calling the informed group. And what changes is that this is their sort of default every morning. We go out with this, you know, this is what we do. And then this is a reflection of having been exposed to the temperature and humidity. Now I know whether to increase my susceptibility. Oh, it's a glorious day. This is a good day to be. I don't need as many interactions to go out versus this is a hot, dry day. Forget it. I'm not going out, or I'm only, you know, I need a lot to get me to go out, right? But everything else is the same. So now I also am going to add this is also a simplifying assumption, because sometimes ants actually do go downstairs and sort of step out of the whole foraging story. But we're going to just look at the story where we fix the total number of ants. So I have 4 parameters now: N, Cu, and Ci, and D. Right, so this whole story we tell with those 4 parameters. And so here's the hypothesis, is that the difference among the colonies and their response to different environmental conditions are the result of differences in how they assess humidity, and how this influences their response to interactions with incoming foragers. Okay, so she's actually done some work on neurophysiology of the biogenic amines, such as dopamine expression in these ants, and so it's possible that there really is a sort of a biological explanation, because of course everything I've told you is a sort of phenomenological model. I'm using a neuronal dynamic to describe ants, right? Which actually a lot of people argue is reasonable, but of course it's not, biophysical. But if there is this connection, it would be pretty useful. Okay, so this is the plot I showed you before, right? Where in essence we just had one C. You go out, you come back, and you're still responding with the same kind of susceptibility. And then here's what happens, and this is just sort of a cartoon, what happens when I have two different groups. One is the green group and one is the black group, and both initially use, this 5 is their initial value of C. They're uninformed, and it's greater than their informed, so it's a hot day, but they assess it differently. So in this case, they're going out along the pink curve, which is the C = 5 curve, and the green group assesses it as, okay, it's a little hot and dry, and they switch to a C = 2. It takes a while, right, because everybody has to get exposed, or many have to get exposed to really see a difference, and so you see it eventually equilibrates on this line. But this group has assessed it as being super hot and dry. This is the kind of day we don't go out. We have such a low susceptibility that you get that kind of, we'll call it like a loop behavior. You go out and come in, like that bottom group in the data, right? And sure enough, just with these 4 parameters in this model, we were able to represent all the data that we saw. I mean, here are the 3 data sets that I showed you on that second slide. And we weren't trying to model every ant at every moment. We're trying to model qualitatively these different kinds of behaviors. And so, here's, with just these 4 numbers, like remember this was the same colony on 2 different days. So they have, we modeled it with the same uninformed C and the same D, time spent outside foraging, but different assessments and also different total numbers, because that can vary as well. And then here's the group that so we can find 4 numbers that give us this kind of loop behavior that's representative of what we saw before. So I want to draw it like this because, well, you'll see why. So if I plot then, this CU versus CI. Remember, this is the default foraging susceptibility, the informed susceptibility. C star is that critical number below which nothing much happens. You can see you have sort of 4 qualitative kinds of behavioral regimes. So the first one is the sort of trivial foraging. You don't go out if you don't think it's going to be nice out, and then you find out it's not nice out. So if you go out and nothing basically happens, right? This is that loop foraging where you have a default that's higher than C star, so you go out, but you quickly find that it's not nice out. So you drop your CI below the critical value and you come back in. Then there's just sort of a steady state foraging, right? You go out, you think it's nice out, you go out, it's nice out, and you stay out, right? And that's what we've seen. And then this one I actually didn't show you. We have other data sets that are well explained, and this is the group that you're kind of wondering what's going on. They're not doing much, and then boom, they all start coming out. And this is well explained with this idea that they have underestimated how nice it is, and they go out, and it's like, whoa, this is a day we really should be spending outside foraging. So they've increased their susceptibility, and sure enough, we can explain that. So here you see the susceptibility has gone up from 0.9 to 1. And we can explain, so this was pretty exciting that we could explain all of these behavioral regimes that we saw and all of the data over collecting it for 3 years, right? But then we started asking these questions, and actually Deborah started painting ants and trying to understand, was there individual differences within colonies? Were some, could we first of all calculate, if some were spending more time, were some making repeated loops, and if so, how did that differ? And so, our model is this kind of population model. Can we then explore the role of individual differences? So And then also, how might this extend to just other scenarios? Just the sort of this basic hypothesis that it's this first exposure to an environment and a change in susceptibility as a result of that accounts for these different kinds of qualitative behaviors. And so it seemed natural, because we were thinking about this as an infectious process, to think about epidemiological models. So the first step was to think about how am I doing on time? Epidemiological models, but with this notion of susceptibility that changes after first infection, right? And so it's a generalization of an SIR or an SIS type model. So for those of you who aren't familiar with epidemiological models, they're these compartmental models where you assume that every agent has one of three states. They're either susceptible to an infection, they're infected, or they're recovered. And the dynamic sort of goes like this: a susceptible meets an infected, and with rate beta, they become infected. An infected recovers with rate delta. What we were interested in is, what if a recovered, so you've already been infected, then meets an infected, they might become infected again, but it could be with a different rate. We'll call that beta hat. Okay? So it specializes to what's these sort of traditional models. SIR is when beta hat is 0. So you're recovered, you have full immunity, right? SIS is when beta hat is equal to beta. You just become susceptible again with the same rate, right? And those have been well studied, and they're super useful. But we were interested in, so it's called, the model actually exists. Nobody had sort of studied it analytically, but we found some people using data to try to understand, and also some numerical simulations. Called SIRI, because the recovered can then become infected. And so we get these in between, the partial immunity, right, where beta hat is less than beta. The ants, it's hot, we're going to reduce our susceptibility, but not necessarily to 0. And then the compromised immunity, which is actually pretty interesting, and people haven't studied that really at all. And and it's actually going to account for that, kind of that delayed reaction, right? And it actually gives us something called resurgent epidemics, which is actually pretty interesting in all kinds of settings. And so here are the dynamics to describe the SRI model. So this is literally, it starts with this well-mixed population, and XS, very large population, is the fraction that are susceptible at this point in time. XI is the fraction that are infected, XR are the fraction that are recovered, and those add up to 1, right? Those are fractions, and so you can plot the state on, this is called the simplex. So when it's right here, everybody's infected. When it's right here, everybody's susceptible. When it's right here, everybody's recovered. Like on this line, nobody's infected. Like right here, half are infected, half are susceptible, half are recovered. In the middle, you have a third infected, a third susceptible, et cetera. And this is showing you how like a susceptible meets an infected with rate beta, they become infected. So this goes up and the susceptibles go down, et cetera. But we also have this reinfection going on, and then the recovery going on. And I mean, what we can do, so in the SIS and SIR models, you can talk about the governing behavior between, infection-free solutions and epidemics and endemic solutions in terms of what's called a reproduction number, which is really just the ratio of the infection rate to the recovery rate. So if it all has to do with like beta is greater than delta. So if this reproduction number is greater than 1, you can expect bad things to happen, and if it's less than 1, you can expect good things to happen. For us, we have 2 reproduction numbers. One is that one, and one is the one associated with reinfection, okay? And, this is straightforward enough so that we can actually analytically, compute like the isolated endemic equilibrium, which is like a steady state of infected population, and we know what that is, and we know what it requires. It requires this one to be greater than, that reproduction number to be greater than 1. We can also talk about what happens on this infection-free, like there's a part of it that is going to be stable and a part of it that's going to be unstable. So this is where nobody's infected, and we can say conditions under which what will happen and where this will be in terms of these reproduction numbers. So everything sort of boils down to the reproduction numbers, which is pretty useful. And so in fact, you can again categorize all the kind of behavioral regimes, right, now in terms of reproduction numbers. So here's the infection reproduction number, and here's the reinfection reproduction, and the critical values are 1, right? It's those ratios, one greater than or less than the other. Here, it's infection-free when they're both small. Here, you get an epidemic, right? You only get, so this is when beta had it, like, along this line is SIR. You can only get infection-free and epidemic because beta hat is 0, right? Along the diagonal is SIS because you can either get infection-free or you get this endemic solution, right? We get all 4 of these regimes because we get the whole plane, not just those 2 lines of parameters. And what I kind of want to point out is this is very similar to these behavioral regimes we just saw. This is like our trivial foraging, our loop foraging, our steady state foraging, and that delayed foraging, right? But I want to just point out, this is super interesting because you don't only, there's sort of some critical initial condition, like if you start with some susceptible and some infected, then there's a critical value. If there's enough infected, you're going to get that resurgent epidemic. And it's sort of interesting, you can see it sort of comes close to infection-free. Looks like everything's fine and dandy, and then boom, you get this really delayed, resurgence to an endemic solution. We call it a resurgent epidemic. So I just want to sort of show you that again, analytically, we can compute everything, that initial condition in terms of the reproduction numbers, so that you know that like the critical value of your initial condition and when it will lead to this resurgent epidemic. Here's just a little simulation for some particular values where we're above that critical value, and the red is the fraction that it's infected, and it looks like it's at 0, right? You might think you've combated it, but then boom, you get this infection. Okay. Okay, so that's already, I think, kind of an exciting story and a way to generalize what we saw with the ants. But I motivated that by saying, well, we want to talk about network structure, we want to talk about individual differences. So we went on to think about that whole story in a network setting. And so there are ways to approximate this whole kind of compartmental model on a network. It requires making some assumptions about statistical independence of these actors. But now you can imagine that everybody has their own probability of being infected, susceptible, or recovered. And then all these rates could be different for, if you're excited about something, I might become more excited about it, or might become less excited about it. And so every agent could react differently to every one of their neighbors in principle. So you get all this heterogeneity. We could all have different recovery rates in principle, right? And so you can write down these equations look very similar to what we had before, but this is for the jth individual in a group of n now. We can have a finite number of individuals, and this is an individual's probability of being susceptible, infected, or recovered. Same kind of idea here, but now we want to collect all those rates, and we collected it in a matrix, a big array of how everybody is responding in terms of increasing or decreasing their susceptibility. So there's an infection one, there's a reinfection one, there's a recovery one, and then we define, now we have 4 reproduction numbers. And they're scalar numbers, and this is called the spectral radius of this matrix. But essentially, they're accounting for all of this heterogeneity and network structure with these 4 numbers. And again, we can analytically figure out these different kinds of regimes in terms of these 4 numbers, which is pretty cool. And in the case where everybody is getting everybody is getting compromised immunity, we can actually just plot it in terms of 2, and we get these different kinds of regimes, and we understand all the geometry of how this can happen. But I want to just sort of conclude by showing you what this then means if, like I mentioned, control strategies or interventions, right? It comes from the fact that we've sort of boiled down these behavioral regimes in terms of just a few numbers, right? These 4 behavioral patterns that are taking into account network structure. So here's just an illustration of what one might want to do. So here's a teeny little network of 4 agents, right, of 4 individuals, right? And the red one is going to gain partial immunity in response to its neighbors. So when it gets reinfected, its susceptibility will go down. the green guy is actually going to have compromised immunity. Its susceptibility is going to go up, and when it gets reinfected, the blue ones aren't going to change at all. Okay, so here's a network, and it's wired according to these arrows. So they're affecting, coming into contact with each other according to these arrows, and we can actually compute based on all those betas and deltas what the regime is. It turns out they're in that endemic regime, the one where they do the steady state. Well, there'll be a steady state infection, and you can kind of see it, that everybody has some probability of being infected at steady state, right? So then the question is, what if we didn't want a steady state infected, right? So here's an example of where we can use our theory, because there's lots of different ways to change those reproduction numbers to put it in a different regime. So this is an example where we're putting it in the infection-free regime, and we're doing it just by changing this guy's, we're just increasing its recovery rate. One guy in this network, and we know, like this theory predicts that should put it in the infection-free regime. This is the same initial conditions, and sure enough, we're in the infection-free regime, right? We could also do it with changing a reinfection rate. So here we've changed one of these reinfection rates and brought it down a little bit. Didn't bring us into the infection-free, it brought us into the epidemic regime, but at least at steady state we have no infection. We could also do it just literally by rewiring. So all we did was cut the connections between 4 and 2 and have it institute those connections between 4 and 1, and that also brought us in the epidemic regime, so it died out. We could also do other kinds of things, like suppose you had some kind of network. So here's kind of a messy network, and here the dark, the medium gray ones don't change their susceptibility when they're reinfected. The light gray ones gain compromised immunity. They become more susceptible, but the darkest ones get partial immunity when they get reinfected. So you might ask this question, who do I expose to the environment, or who do I vaccinate? Who do I, who should I pick? Suppose I only have a certain amount of vaccination, I can only pick a certain number. Who should I be such that it'll put me in the right regime? This is it's in that bistable regime, the one where we get this resurgent epidemic. So the black line is where we've done nothing. If, this is just to sort of show you, if we picked agents 2, 3, 5, and 7 to pre-expose, it actually doesn't do very much, right? If we pick just agent 11, it does a huge amount, right? So what we aim to do is to get tools that you could think of this as like a centrality index, a nodal index that tells you how much influence that individual will have if we change their properties, right? So sure enough, 7 and 11, does better. And then if we do 7, 11, and 13, I'm actually not sure if we're out of this regime or if it's going to resurge later, but at least we have a lot more time to handle the fact that stuff is going on. So just to conclude, I sort of hope to convince you that using analytically tractable models allows this, I think, pretty powerful way to investigate systematically. So looking for mechanisms, working with data and models together to help explain. In this case, we were looking at resilience and spreading. Some of the key features were this, hypothesis about adaptive susceptibility after first exposure to an environment. I didn't really get into it, but what was really at play in these models is sort of these different negative feedbacks, which is associated with regulation, positive feedback, which is associated with sort of destabilizing, but also switching and responsiveness to environmental cues, especially when they're at different timescales. Also thinking systematically about role of interconnection topology and heterogeneity, which can show up all different ways, at least in the animal world. There's personalities, there's your state, you might be sated, you might be hungry, there's sociability, there's skill, you might be fast, you might be slow. And then the generalizability allows us to move away from the sort of the model organism to others and think about testable hypotheses in other species, and also to design strategies for control or enhancing spreading. And this goes for technological systems too. So thinking about how I want robots to respond to environmental signals. They're doing like search and rescue. What if they hear something? What should they do? What if they have very limited means to communicate? How should they be responding? So here's just a little example. So we've been we started thinking about this in the context, obviously, of ants, but I've been working so long in fish, and we're really interested in these ideas of like what happens when there's a threat, and some of the fish are the calm ones, and some of the fish are the agitated ones, bold and shy. There's all kinds of things. What if you have a positive experience, like, oh my God, that was so scary. Next time I'm not going to wait for so many others to do it. I'm going to go with it. Or what if it was negative? It was like, that was a super waste of time. That was not scary. Or likewise with food. So here's just like a little from Ian's group looking at the different color fish. So this is a fish tracking experiment where some have been trained to be responsive to, I think it's like a little laser dot or something that triggers them, but then the others are just responding by the social cue and thinking about how that works. We're also thinking about things like multi-layer networks. This is sort of like the sensor fusion problem. So fish in particular, they have the lateral line, so they can sense, like the set of neighbors that they can sense like through unsteady flow is one set of neighbors that very close. The visual network is more like a body length away. It's also olfaction, so then there's even further away. So we're asking questions like, what if these guys, the ones that are close by move, but the ones that don't, the ones you see don't, are some going to be so agitated that it's like an or. Either these guys move or these guys, I'm out of here. Or are they like the calm, wise older fish that says, I'm so happy, I'm not budging. I've seen this before. I'm waiting till both groups, move. And then what about like role of individual differences? If we have a composition of like some ors and some ands, does the group as a whole do a really good job of responding to real cues, but rejecting false positives and disturbances? So these are the kinds of things that we think about. So thank you. I just want to thank my research group. There's Renato and my family and my funding. And thank you.
Great, very nice. Lots of questions.
Sorry, the slide I showed where you...
we have to use the...
Oh, sorry. I'm sorry. I gotta rethink my question now. So in this experiment, it's interesting. Okay, you shine the laser, a subpopulation of fish will react to it and move. So how do you discriminate? Like, why? How's the system robust enough so that one rogue actor would not drive the whole pack to disaster? Because do you understand what I mean here?
Yeah.
Because
I mean, they do this experiment sometimes with just like startles, like nothing's going on in the tank, and somebody will just startle, and they look to see under what conditions it might spread or not. So this was a sort of a manipulated where individuals were trained to respond to something, but.
I'm saying, let's say, like you have kind of a mutation that renders one fish crazy in a school of fish.
Yeah.
What would prevent the whole school from following that?
Well, this is what we're trying to understand. In fact, Ian is working with somebody at Harvard who is looking at mutations of fish in their social skills or their social ways, right? So like even like sort of the autism spectrum for fish. So how do you how can we think about these different mutations that might reflect somebody ignoring the others, or somebody over responding to the others, or swimming really differently, and then thinking about what happens when you put together different compositions, or introducing just one mutation one mutated in a group of others. This is what we're trying to unpack, and what we're trying to do is try to answer those kinds of questions, like under what conditions is it just a disaster? Are you like responding to noise and perturbations? You can't what do you do to this? What happens when the starlings just can't do it anymore, right? They just like explode, right? What explains that? And so people do these experiments where they try to insert like fake fish, it's very hard because accepting them as conspecifics is really challenging. But this is where models can be super helpful. So you can come up with testable hypotheses, but you got to go back and forth. You got to see what you can do with the experiments and what you can do with what the models say, and then you work towards coming up with hypotheses. In fact, Deborah was always wanting to come up with hypotheses that we could disprove. So what we try to do is come up with ways to explain like all the possible things that can happen, and then show that these can't happen, these can't happen, these can't happen. So we're getting closer to like providing more evidence for the what remains. I mean, these are other ways to think about handling it, but yeah, those are I think super important and challenging questions. But
Hi, I had a question about the anthills. So you could measure stuff for the same anthill again and again and again, right? So I was curious, you had these 4 parameters, C and D,
Yeah.
and you would imagine that, if they lowered their C, so they're ready to go out, they might actually stay out longer. So these parameters could correlate. So if you measured the same anthill again and again and again, in these 4 parameters, do you see sort of, relationship between them? Are they independent? Are they really just one
Yeah.
parameter where,
I think that the amount of time that they spend outside, they're willing to spend out, it seems pretty fixed for a particular colony, right? It's sort of, I mean, some of them go like 20 meters. They'll go really, or they'll go like a long time before, it's sort of like the goal is just to find a seed, but I think and it could also have to do with where they are located relative to other ones, or, but my understanding is that, that's pretty fixed. It's that N that's going to change, like, so we fixed it. The numbers can be very important, and I think she knows less about it, so she's done these, she takes the dirt off the nest and puts down like a piece of plexiglass with a camera. It's so painstaking because they build up by the next day. It's all covered up again. And it could be that day the ants are just like, they don't even come out, or, so,
the hidden variables which you don't know.
Yeah, it's hard. So she does have some data of ants, but it's very hard to see inside. And once she tries to move things to the lab, everything changes because you're not in the real world. And yeah, there was.
It's really interesting how you can use the models to reflect what the data shows.
Mm-hmm.
Do you try to do this to the numbers, or are you only interested that your models represent the qualitative behavior? And
Yeah.
is there value in the numbers? And then do you do something that typically is done in machine learning, that you have training data and test data, so you leave out some data and then later test if your model can explain this data?
Yeah. So yeah, we're not trying to do at the level of like numbers and individuals. We don't. Yeah, that's sort of A, it's kind of hopeless, and B, it's not so interesting, right? We're trying to understand the mechanisms that explain these different kinds of behaviors and that would distinguish this sort of key feature that seems tied to fitness. And so for me, those aren't the interesting questions, like to figure out where every fish is at every moment of time, but what drives these kind of behaviors, like responsive versus robust, and those kinds of questions, in some sense, are more qualitative, right?
And I have one follow-up question. So this infection model, what is the connection to how opinions spread?
Absolutely. No,
Is there one? I don't.
There is one, yeah, absolutely. So, yeah, people have used these epidemiological models and using them more, like spread of misinformation, these kinds of things. In fact, there is a whole literature we discovered that we actually referenced in our paper, from the 50s in psychology, called inoculation theory, which is literally just adopting epidemiological stories. And it's kind of interesting because they do have this whole, it was a recent paper, like a 2019 paper, where they did some experiments with human participants to try to bring evidence to this theory that once people have been exposed to like fake news or misinformation, it's really hard to get them to change. But if you can kind of reach them beforehand, inoculate them by showing them, sort of some examples of the the root and the consequences of misinformation, or how to spot it and what it means, then they actually will be less, this is sort of like, they do this with topics that aren't like hot button topics, right? Just to see if people react. And so, yeah, absolutely. Like all these, like you can find lots of papers where they have like these curves. In fact, we noticed that people are having trouble with that delayed epidemic, right? They basically put in a delay, right? Whereas our model would predict this kind of resurgent epidemic in like an information passing story.
Great. I think with that, in the interest of time, we'll wrap it up.
Okay.
Thank Naomi again for a great seminar.
Thank you.
And I think we have a wee gift,
Oh,
so you can remember us.
you.
And I've been
Ooh.
told you shouldn't open it because it's fragile, so you don't want to disrupt the special packing.
Okay.
Okay? So
Thank
thank you
you
very
very
much. Okay, bye-bye.